So we're going to be looking at the structure of cell membranes. In terms of understanding, first of all, what a cell membrane is, we need to take a look at where they are found. So we start here with looking at a bacterial cell or a prokaryotic cell. And this prokaryote or bacterial cell has DNA in the center, and it is not wrapped up in any kind of membrane or nucleus. And instead, the only thing you have is a single membrane, which is going to be right there. Okay, so the whole thing is wrapped up in one membrane, which we can call either the plasma membrane or the cell membrane. And many times bacteria are also going to have a cell wall that then surrounds that membrane. Here we have two types of eukaryotic cells that should be fairly familiar. On the left, you have the animal cell, and on the right, you have a typical plant cell. And there's a couple of things that are going to make eukaryotes a lot different than prokaryotes, namely the fact that they have a nucleus, so pro, no, euc, nuke. But that's kind of a more basic way of taking a look at these cells, and the biggest thing about them is that eukaryotes have membrane-bound organelles. So all of the different membranes, all the different organelles that are within the animal and plant cells, as well as fungal cells, as well as various other types of protist cells, all of them are going to have organelles that have membrane around them. And we can take a look at exactly where these are found. We can start by just taking a look at the animal. So we have a membrane that is going to be surrounding the nucleus. And then we also have the endoplasmic reticulum that is all made out of membrane. So all this material right here. Bloop. Right? So that is our rough ER. We know because it has ribosomes or little dots all over it. We've also got the smooth ER over here. So everything highlighted right now, all of this is membrane. So the ER then continues on to the Golgi apparatus as part of the endomembrane system, and that's going to pinch off and make a whole bunch of little vesicles as such. We also have the mitochondria in animal cells, and those too are made out of layers of membrane. We've got an outer membrane, an inner membrane in there. If we take a look over on the plant side, a lot of the same things that we just saw. And the plant cell is also going to have a couple of things that the animal cells do not, namely a large central vacuole right here, which is, again, made out of membrane, and some chloroplasts, right? We've got these guys right here. They've got the outer membrane as well as the thylakoid membranes kind of filling up all those spaces right there. And of course, just like all cells, both animal and plant cells, in fact, all types of eukaryotes, just like what we saw with prokaryotes, all cells have a membrane that kind of surrounds them that goes the whole way around here, right? They separate what's going on in the cell from the environment. That was a horrible tracing job, but that's all right. An important thing to note, of course, is that with the plant, you also have a cell wall, just like we saw with bacteria, although the cell wall is made out of something different. So we can take a look now and start kind of zooming in at exactly what the membrane looks like. And that is this right here, kind of this double layer of various chemicals that are all interacting together, and some things that are getting embedded within that. So keep that in mind as we're taking a look at all of these membranes, everything that I highlighted here as well as the outside. All of that is made out of this type of material. So here's the membrane a little bit more on its own, so it's easier to see. You can tell that the major component here is going to be the phospholipid bilayer. And this acts kind of as a place for everything else to be embedded. And it's also going to be selectively permeable, meaning that only some things can get in through it while other things are locked out. In addition to the phospholipid bilayer, which, as you can guess, is made out of two molecules by two layers of phospholipids. We'll talk about a phospholipid. In addition to that, we're going to have some cholesterol molecules, and cholesterol molecules are going to kind of mainly just squish on the inside of this sandwich right here. So the cholesterol is going to be what is responsible for cholesterol, keeping the membrane nice and fluid, as well as giving it some integrity, keeping it nice and not rigid so much, but preventing it from falling apart. We've also got all kinds of proteins embedded in the membrane. In fact, that's going to be a huge part, are all of these various proteins. And those proteins can go all the way through, like what we see right here, or it can only be embedded in one side, either the inside or the outside. So the ones that go all the way through, these guys are called the integral proteins, and the ones that are only on one side are going to be called the peripheral proteins. And regardless of where they're located, they have all kinds of different functions, kind of like when we looked at proteins in general, just tons and tons of functions going on here. They can act as enzymes. They can also act kind of as channels to allow things through. They can also act as anchors, and they can attach to other things. So what we're taking a look at here up at the top, sorry, I'm just getting a new color. These guys right here are some of those other components that, in this case, these guys are sugars that get attached to the outside that just signals to other cells what type of cell this is right here. Okay. So finally, as we said, the phospholipid bilayer is made out of 2 layers of phospholipids. So we need to take a look at exactly what a phospholipid is. Phospholipid. So your phospholipid is made out of 2 parts, a phosphate group at the top, bada bing, and lipid chains at the bottom. The phospholipid has kind of this dual characteristic, 2 different properties depending on what you're looking at. The phosphate, because of the fact that it's got ions there and it's very, very polar because the oxygens here are pulling electrons more closely towards it. So this whole thing ends up being very polar, very ionic, very charged. And so when you get other molecules, such as water, in around it, those are attracted. Yay, hydrogen bonding. And so this area, we are going to say, is very, very hydrophilic, or water, hydrophilic, like Philadelphia, meaning city of brotherly love. Philia means love. So hydrophilic is the portion of the phospholipid that absolutely loves water. On the flip side of that, the lipid with all these carbon and hydrogens up in here are really non-polar. They do not attract well to water, just like any other lipid. They are hydro, meaning water, phobic, meaning fearing. So these guys don't mix with water. And so when we were taking a look at the phospholipid bilayer, the fact that you have 2 of them, the reason that occurs, sorry about that, here we go, is because both the inside and the outside of the cell are surrounded by water. We've got all of this water out here and in here, and the hydrophobic tails right here are repelled by the water. So they're going to orient themselves inward, kind of getting sandwiched in there, while the hydrophilic heads, these guys here, are interacting and they're fine with the water. Okay, the next topics that we're going to look at are, how do materials get through this membrane? When we're looking at any kind of homeostatic mechanism, you have to have a way to allow some materials to get in and out while blocking others. If you just let everything go on through, you're going to have some havoc. You'll find out why today. So the next 2 chapters are all about how materials get in, get out, and how they're related to homeostasis. You're going to start today with looking at some passive transport.